Hi, my name is Erick Moen. I'm a postdoc fellow in the Van Valen Lab here at Caltech. I want to take a moment to thank the workshop organizers and the committee for giving me this opportunity today to talk about our work really over the past couple years. So as you can see, the talk is entitled Machine Learning Infrastructure for Single Cell Biology. So we're going to discuss our automated approach to data annotation and model development, basically how we transform biological image data from multiple experiments into standardized training data for deep learning. We'll also cover a little bit about what we hope this will do for all deep learning enabled biology in the near future. But before we look ahead to the future, let's take a look at the past. So the gentleman on your right that you're likely familiar with is Johannes Kepler. He developed the laws for planetary motion, as you might remember. But he didn't develop those laws in a vacuum. He didn't walk outside one day, look up in the sky, and start writing down mathematical equations. Instead, he based that on a lifetime of observations, of measurements. Those measurements were given to him by Tycho Brahe there on the left. He was known for not only the quantity, but the accuracy of his astronomical observations. And when he died in 1601, he left this compendium of knowledge, all these measurements, to Kepler. And then Kepler was able to take that and extract the knowledge and insight from them. In many ways, you could say that we're in the Tycho Brahe era of digital biology today. Why might you say that? When we look at the current data building efforts that's going on within the biological community, and as I mentioned there, this is by no means exhaustive, there are massive movements in gathering and cataloging data. It's funded, well funded by NCI, NIH, private organizations. It includes multiple organizations, multiple universities, consortia, and research centers that are not only developing new ways of getting this data annotated and collated and organized, but then be available through software. And I've mentioned just a few here, but all of them focus on different technologies like seq, codex, fluorescence, light sheet, and how our bodies deal with disease or go through normal function. But there's never before been this sort of explosion of data across all the different realms of biology. And this is occurring at a very interesting time for the computer science community, where the advent of deep learning is sort of coming to dominate the field. It seems like every day we see new approaches to old problems like image restoration, denoising, segmentation, tracking. Deep learning holds the promise of doing all of this for us, but maybe even more than that. Given all the data that we have, deep learning could not only mine that data, but add to it. How so? Well, let's take a look at just one example. This would be whole cell segmentation. If you look there at the left, this is an image, a composite image from a multiplex ion beam imaging scope. Not so much important about what that technology is. Rather, let's focus on the idea of how we would extract information from that. So right now, even with some of the best deep learning techniques, what you do is try and find the nuclei and then do a simple expansion. But what do you lose when that happens? Well, this technique, imaging technique, is designed to bring in as many different channels of data as possible. And as you can see there, if we only focus on doing that nuclear expansion, we lose a lot of that information. So instead, it would be ideal if you could have something like whole cell segmentation there, where you could do quantification across those channels. Not only that, then you get that you get marker quantification, you get subcellular localization, and you could do some morphological analysis on top of that. So if we do segmentation well, then we immediately improve the data store that's available to us. So given that, what's preventing deep learning from broadly delivering on this promise? Well, quite simply, it's hard to standardize biological image data for one machine learning task, let alone for deep learning generally. So again, let's stick with this instant segmentation in tissues and just look at the raw data from that previous multiplexed image. So this is just 4 samples, 4 slices of tissue, 1 disease type, 1 imaging platform. And yet each of these samples are going to have different expression levels. Just to compile the data set to learn on, you'd need to pick the right channels, normalize, resize the images, and so on. And we haven't even gotten to the model yet. Well, okay. Let's try and make this a bit simpler. Let's just look at nuclei on a standard scope, right? Even then, the possible variation in image sizes, resolutions, cell types, marker expression, frame rates, they'd all need to be handled before you were ready to try your first model. So it's not just that we need more data. We need a holistic way of processing and preparing that data so that it can be useful for biologists and their explorations, as well as for machine learning researchers who may develop the models that then provide that insight to biologists. And that's really where we begin our story. We hope to develop a system, which we're calling DeepCell Foundry, where biologists who study different things from cells to tissue, even whole organisms, using different methods, could contribute data and hand that data off. And in return, they get some bespoke analysis software that would help them do their analysis. On the flip side of that, the other side of the image, as you see there, you have the machine learning researchers who could then get some real world data that would be well formatted, and then they'd be able to go on and develop various theories or algorithms that they could apply to different models, maybe even outside the space of biology. Rather than try to convince you, though, that we found some sort of generalized solution or generalized method that's successful in all the different cases that we need it to be, I just want to walk you through two specific examples that we've worked on that illustrate the steps that you'd need to take to make this sort of approach general. So let's return again to our simple exposition of segmentation. Now, I said we were going to start simple, but even if we ignore the normalization, image sizing, marker selection, expression, all those things, we still have to figure out a way to turn the left image into the right image, because every supervised learning technique needs high quality annotated data, and the data has to come from somewhere. So in days past, that meant someone, usually whichever student was taking the lead on the project, would have to sit down and circle some cells. Now I know this because I was one of those students sitting down and circling cells. So let's say you get this first image done and you're happy with it. Awesome. Now you have to hope that every other image looks close enough for that to be valid. And what you're seeing here, even with our simple whole cell task, when we start branching out to different platforms and different cell types, that's just not the case. And remember, you're going to need markers, or you're going to need labels for both nuclear and whole cell. And in some cases, you'll see that the nuclear signal isn't there, and yet you'd still like to try and predict it based on the context of that whole cell. So what do we need just at a base level to get this project started? Well, obviously we need some way to create labels. We have a couple of requirements, we think. We'd like to be able to correct and create annotations and do this in a collaborative fashion. And we know that we're going to be working on multidimensional data and lots of it. So we need to be able to handle that as well. And that was really our first project. One of our first projects in the lab was building an annotation platform. We call ours DeepCell Label. The sort of tagline there is that it's browser-based software for distributed labeling of biological image collections. Now, I say distributed because it's crowdsourceable. And what that means is that we're able to bring non-experts in to help accelerate biological discovery. Because otherwise, what we're doing, even with this great platform that's custom built to a specific machine learning task, it's still one individual sitting down and doing that task, circling those cells, for instance. So how do we make that better? How do we go faster? Well, in our case, the key to our approach is human in the loop. We have crowd annotators that will eventually be correcting model errors rather than trying to make these images from scratch. So the process works like this. From left to right, what you'll see is the initial annotation. So we will require some experts with some knowledge to go through and provide us with basically a bootstrap data set. We can take that data set and start training a model on it and get some elementary predictions on new data. And that's what you see there in the middle. Then we start writing up some instructions that help us understand where maybe the model made some errors, Maybe if we've been through this passage a few times where annotators and models tend to make some errors, and we go through and have them correct that in an iterative fashion. And then when you get to about 10,000 labels or so, what you'll find is that you've reached that third area. You have a trained model that's good to go through and get as many annotations as you'd like. And I say that kind of leveraging our end result, TissueNet. And what you'll notice there on the left is what we've done with this sort of human-in-the-loop system with our label annotation platform is that we were able to build a data set that was an order of magnitude larger than the rest of the field combined. And that really is leveraging the power of using these models and this crowd annotators together. And we can then start branching out from not just single images and single platforms to multiple platforms, multiple cell types. And you see there, we're still able to keep that expert time that's having to be put in by, let's say, a single student on a single project to a minimum as compared to what the crowd's taking on. So how do we perform? Well, you can see a few of the model outputs here. I'm going to save the F1 score for the paper that I'll show you on the next slide. But just as a cursory glance, if you look on the top, we see that we've done quite well in being able to generalize across different platforms, which you see there in the brackets, as well as different tissue types. And just to give a little bit of a plug, because I know I glossed over some of those performance metrics, you're welcome to either look at that paper that I've cited, or we do encourage you to go to deepcell.org or any of our GitHub pages. And what you'll find is that we've provided several different avenues for you to go through and use this model, as well as go ahead and take a look at the data that we've developed that really powers this model. So just a small plug there. But kind of getting back to that theme of data, one thing I wanted to kind of bring out is that we have human level performance using this TissueNet and our models. We get human level performance with that. And that's particularly interesting when we think about the fact that the ground truth is a little murky, right? In some of those cases, just for instance, we didn't even have nuclei. And yet, whenever we compare our segmentations to an expert segmentation and have a pathologist kind of come in, a trained pathologist, and decide which one they like better, it's really hard to tell the difference. So we achieve parity there. And I don't say this to really brag about our model. And as much as I'd love to say that we have best architecture and so on, what it really comes down to, and what I really want to stress for this talk, is a truism in deep learning. And that's that data is ultimately all that matters, or it certainly is a large driver of performance. And to kind of bring that point home, if you look here at the pre-trained models that we have, especially Stardust and CellPose, which are sort of cutting edge, even those really well-performing algorithms, whenever they're given a TissueNet to train on, which you see there on the left of that image, you'll see a pretty dramatic boost in performance. And so it really is the data that we've made available in this process of preparing and creating the data that can really power all of the really good deep learning solutions that are already available in the literature. And that kind of brings us to our big question, right? Can we automate this process even more? Can we generalize it to different machine learning tasks? Well, to answer that question, let's walk through our other sort of simple use case, cell tracking, as you see there in the lower right-hand corner. So again, just to state our concrete goal, what we're hoping to do really is not so much focus on the algorithm, but the data set. We want to create what we're calling live cell net, an image net for live cell imaging. And so really key here is getting both nuclei and whole cell segmentations or annotations and connecting those over time, taking special note of the divisions. And just again, to kind of drive the point home, we're not the only ones trying to develop cell tracking and deep learning, though we very much like our approach and our algorithm. We're very hopeful that no matter what happens, having this underlying data and getting it to scale the way that we did with TissueNet will improve all of the algorithms that you see here on this slide. So let's take a look at our approach. This time we formalized it. And by that, I mean that we've tried to automate as much as we possibly can. And take note of the arrows here, because each one of those arrows, or at least most of them, when I say automate, it means that we can run that process from really a single command in a terminal. So let's break this down piece by piece. We're going to start with what we've known before, which is the annotation pipeline. And again, we're going to take DeepCell label, and this time we've abstracted sort of the underlying machinery to make it that much more customizable. So instead of just having it work on segmentations, now we have some task-specific tools, or in this case, tracking-specific tools and alerts that allow us to link up various lineages over time. And there we can use our same human-in-the-loop approach, this time writing slightly different instructions at each go-around to maybe be tracking specific of how you link, how you say what cell belongs to what lineage or what daughter belongs to what parents and so on. And we sort of run that through, and we're able to scale up our annotation throughput in much the same way. Now, the big difference, or the key driver here, is from our sort of previous method, is having the data registry. Now, what you'll notice again with these arrows is that from the time that the data is collected, it is being tagged, it is being versioned. So whether it's collected and dropped into our data registry, whether it's sent out for annotation, when it comes back, when it's QC'd internally, at each point, we have a record of everything that happened to it. We have a record of all of the metadata associated with the image, as well as maybe even each of the crowdsourcing jobs that we've launched. So what does that look like in practice? Let's pull back the curtain just a little bit. So for each individual image, we may need to crop that down to size, because if we're sending it off to be annotated, oftentimes annotators like to maybe look at a smaller field of view. So we need to be able to track which crops went where. We need to be able to run that through our model, track which model was applied to which version of the model was applied to each piece of this data, so that if we do need to go back through another cycle, we kind of know where we're at. We know when we need to update a model. We know what segments and what crops have been sent to the crowd and when, and what version is the latest. We use both DVC and Git in conjunction with one another, as well as our storage remote, to sort of manage this entire process. And what this allows us to do is really track the data provenance the entire way. And so if we do need to change something, if we do have updates, we always know what model works where, what it was trained on, and where we are in sort of the stream of time of improving that model. Now, using this, we were able to go through, and while we're still very much in the process of building LiveCellNet, I do want to share with you just a few numbers to give you an idea of how quickly and what sort of scale we're trying to look for as far as LiveCellNet. You can see there the number of segmentations, as well as the number of whole cell segmentations that we have across different imaging types. And just to kind of really bring this point home, since we're still sort of building the data and the numbers are a bit squishy, what I'd like to focus on is really what the DeepCell Foundry enables. And that's that we can do an entire data set's worth of machine learning currency. So in this case, building that data set, we can do that with the cost similar to one sequencing experiment. So we're sort of estimating that, as opposed to TissueNet, which was quite expensive, whenever we're sort of proofing all these pipelines, that our next data set will cost around $20,000. The big takeaway here is that by automating so much of the mundane and sometimes tedious tasks associated with annotation, by keeping those down to a minimum, and by minimizing the amount and the time that lab personnel have to spend on labeling and even just shuttling images around from one place to another, doing those normalization tasks, we can keep the high quality of annotation and really scale up while keeping the costs low. And kind of a last thought there on cost. There's another paper that's been released at the conference just last week. It's Reduced, Reused, and Recycled: The Life of a Dataset in Machine Learning Research. And this sort of drives home the fact that the reason that so many data sets are reused in machine learning is because it's so expensive, both in personnel and in time. And so I pulled that quote there. Dataset development is time and labor intensive, making large-scale dataset development potentially inaccessible to lower resourced institutions. And so one thing that we very much hope that the DeepCell Foundry will do whenever we can finally get everything released and make sure that it's all available to everyone, is that it'll really help the community as a whole flourish by helping labs start their own dataset development efforts, regardless of the resources they might have. And that kind of feeds nicely into this. I do want to record all of the different web addresses for you to be able to go and review. All of our libraries are open as under a permissible usage license. They're all available on GitHub. And so we do very much hope that this becomes a community resource that everyone can contribute to, and it really becomes something that the community can have ownership over. If there are any thoughts or questions, I put my email down here. And again, I've listed a couple different links, including Intro to DeepCell on our GitHub. That would be a great place to start if you're looking to kind of figure out where to go, at least with Mesmer, and then we hope eventually the rest of this information as well. So lastly, I'll go ahead and thank my funding sources, as well as all those who took part and made this research possible. So I look forward to taking any questions. And again, please do feel free to reach out at my email address if anything does come up. So thanks again for your time.